Hi, welcome to BASF's webinar, Unfit for Function, Chronicle of an Unsuitable Batch of poloxamer 188. My name is Phil Butler. I'm part of the BASF Pharma Solutions Pharma Ingredients team, and I'll be your moderator today. Before starting, it's my pleasure to introduce our speaker, Nadya Morales-Cummings. Nadya is the Global Technical Marketing Manager for Biopharma Ingredients at BASF. Nadya supports customer interactions around our biopharma ingredients, as well as supporting innovation activities focused on helping meet our customers' future needs. Nadya holds a PhD from Yale University and has been with BASF for 11 years. During that time, developing technologies and solutions in the areas of industrial biotechnology and biopharma. Dr. Morales-Cummings' webinar today will focus on the risks that a production can undergo when a raw material is not suitable for the application. Briefly, Dr. Morales-Cummings will discuss the negative impact that a regular, non-bio lot of poloxamer 188 had in the production of several biologics during 2021. Nadya will present data generated in the new BASF Biopharma Ingredients Lab on the impact of this suboptimal poloxamer 188 on CHO cells. Nadya will also provide information as to why Kolliphor P 188 Bio is the optimal shear protectant for bioprocessing applications. So I hope I did a good introduction for you, Nadya. I'll turn it over to you.
Thank you so much, Phil, for that kind of introduction. Thank you, everybody, for taking the time today attending. I will go to our next slide. And I actually wanted to ask the audience if any of you who have maybe are part of bioprocessing or have in a team that actually makes a biologic, have you experienced any decrease in cell viability due to poloxamer viability in your production? I'll give you a few seconds. Yes or no, it should be, I hope, easy to answer so we can move on. So I'm gonna go ahead, move on to some of the, so the results are being updated live. And it seems that some of you actually have been impacted by this, by this problem. So let me then begin introducing the topics that I will be discussing today regarding this. So I will start with a brief overview on what is the impact of raw material variability and bioprocessing. Then I'm going to move into a brief introduction to poloxamer chemistry and poloxamers in general. Then I will talk about hydrophobic species as being one of those species that actually contribute to the variability of poloxamer 188. And lastly, I will show you actually some data that we generated in our lab regarding the response of some CHO cells, as Phil mentioned, to a known species in a batch of poloxamer 188. So let me start with the effects of raw material variability. There you go. So raw material variability is part of the risk management assessment that many of you do in your process. And it's actually really important, and I feel that I'm preaching to the choir here, of course, right? Because you know that it can actually have a really big impact. It can have decreased productivity in your process. Sometimes raw materials can have, their variability can lead for processing consistencies, right? When your process, sometimes it runs perfectly and you get the yields that you're expecting to get, and some other times it just completely misses the mark. That, of course, leads to, we're in a highly regulated industry, right? So that actually then starts raising compliance concerns And of course, it actually can affect the final product that you're trying to make, right? So, which is why it is so important that we try to minimize that risk. When I talk about this topic, I always like to refer to this white paper because I actually find it very useful. So the Bioforum Operations Group, we lovingly call it BPOG, is an industry group with the idea of accelerating progress in the biopharmaceutical industry. And the way that the BPOG work is that we actually have different work groups that work in different topics. And one of the groups actually generated this great guide on how to actually help you assess and manage the risk of your raw materials in your bioprocess. It's a great read, and I highly recommend it. So let me tell you now a little bit about poloxamer chemistries and how is it that it works, right? So poloxamer chemistry was invented in BASF. It was invented at a Wyandotte, Michigan site in 1973. And poloxamers are essentially amphiphilic copolymers. What does that mean? It means that it has both it has a part that is hydrophobic and a part that is hydrophilic. The hydrophilic piece is brought in by the ethylene oxide, which is in the light blue. And the hydrophobic part of the poloxamer is brought in by the propylene oxide, which is in the green. And essentially, it is a tri-block, right? And it actually assembles itself with, it is essentially a sandwich of propylene oxide, right? It's sandwiched by bread made out of ethylene oxide. And what actually gives a characteristic to poloxamers and what makes them so attractive as surfactants in many different industries is that by changing the composition, so the ratio of composition to ethylene oxide to propylene oxide, you can actually manipulate a lot of its characteristics, right? So it can be part of oral dosages, so part of the pill coatings, for example. It can also be part of a cream, right? A medicine that is actually administered as a dermal administration, or even cosmetic creams. It also can be part of detergents. So what I want to highlight is that there are many different poloxamers qualities used for different applications. So there are many, for example, in our case, the poloxamer we care about is 188. And there are many different grades and qualities that are manufactured, for example, at BASF, to actually fulfill the requirements of the different industries that it's used in. Poloxamer 188 as a shear protectant is actually a very interesting proposition because it is very much a Goldilocks kind of find. It turns out that it has the perfect ratio of ethylene oxide and propylene oxide to have a really great interaction with the cell membrane. And the way that it works is that the hydrophilic parts of the poloxamer, which are the light blue tails here in my screen, they are absorbed and interact with the hydrophilic heads in the phospholipid membrane. The propylene oxide head of the poloxamer then interacts with the hydrophilic tails of the phospholipid molecule in the cell membrane. And what it does is that it gets absorbed and it gives the cell more robustness without actually increasing the size of the cell, right? So it's a very tight interaction. And there can be other species inside of the poloxamer 188 that is not poloxamer that is not fit for function that can actually disturb this interaction. So that leads me to my next topic, which are the hydrophobic species. These are some that are very well known in poloxamer 188. So everybody here knows, again, preaching to the choir, critical material for shear protection in the bioprocessing industry. And what I want to make you aware is that even when you buy a poloxamer 188 that is pharma grade, and that it meets several of the pharmacopoeias, for example, that does not mean that it's necessarily compatible for cell culture, right? Another thing to consider is that when you buy a poloxamer that is not fit for cell culture, you can actually see variability in your processes depending on the cell type that you use. Some cells are more, can be more sensitive, right? So if there's any variability in the poloxamer, maybe some cells in some of your processes may be more sensitive to that change than others. And essentially, at the end of the day, having a lot of variability puts you at risk of having ultimately a process that is not as productive because your cells are not as viable when they're in the presence of those big shear forces inside your tank. And what we have been doing in BASF is actually investing a lot of resources and actually trying to understand better what is it that happens inside of the tank. So in cases where you have, for example, hydrophobic species as a impurity in your poloxamer, there is a change in the way that the cell is protected. And there is a good reason for it. So hydrophobic species, by definition, are actually bigger than the poloxamer 188, right? So that ratio of ethylene oxide and propylene oxide is changed. And propylene oxide, it's bigger in this hydrophobic species. It has more propylene oxide than the poloxamer. And that actually makes it more surface active. What does that mean? It makes it that it's more, it actually wants to interact with the cell membrane much more than the poloxamer 188 does. And because it's so much bigger, when it introduces itself in the membrane, it kind of destabilizes the membrane. And it disrupts the shear protection that P188 can offer the cell. Because they're more surface active than the poloxamer 188, it actually outcompetes it to get into the cell membrane, even when it is in very small quantities. So that is something to consider. So what has BASF done to actually to try to prevent this? So with our Kolliphor P188 bio product, one of the things that we developed as part of that product is actually a validated reverse phase HPLC analysis. And essentially, it is a test to detect not only hydrophobic species, but hydrophobic species of a higher molecular weight. So here on the left, you can see this is a picture of an HPLC on how it looks like, and it has 4 distinct regions. The first region here is the lower molecular poloxamer 188 species. So poloxamers are polymers, right? So you actually, when you buy poloxamer, you buy, there's going to be a range of molecular sizes, right? So we adjust our manufacturing so we can actually get the right molecular, a higher distribution of the molecular weight that is that people are looking for, which is that second peak, right? So that is the main P188 peak. If they're hydrophobic species, it can be detected in this third zone in the chromatogram. And what you can see here, so these are lots when we were developing the Kolliphor P188 biograde, we actually started analyzing a lot of the lots that customers had actually flagged as being problematic. And we actually found, you can see that different lots have different degrees of concentration of those hydrophobic species. And then lastly, the last part of the graph is that fourth part, which is the higher molecular weight P188 species. Oh, it moved before I wanted it to move. I apologize. So what we actually did developing our method is that we improved. We started with one method. We were able to detect those hydrophobic species, and then improve upon the method so that we can ensure that we have a detection limit of 250 ppm of these hydrophobic species. So when you buy a lot of Kolliphor P188 bio, the specification is a maximum of 250 ppm of these hydrophobic species. And to give you context, when we talk to customers about the effect of these hydrophobic species in their systems. Even the customers with sensitive cell lines, they don't see any effect of the hydrophobic species unless they are in 800 ppm or more. So this analytical method, of course, is a part of our COA and part of our release criteria. We can actually, as I mentioned, we can detect 250 ppm of hydrophobic species in each one of the lots of Kolliphor P188 bio. And essentially, our biograde offers all of the shear protection that poloxamer 188 is known for and has been published about, diminishing that risk of having hydrophobic species that could actually be hindering the viability of your cells, right? And by default, then minimizing some of your product yields. So those are the known. Yes.
Are there other tests for Kolliphor P188 bio that we perform that aren't included with our standard grade pharma poloxamer 188?
Yes, Phil. There are several other tests that we do for our biograde. So we do a total aerobic microbe unit count test, so the TAM&C. We do a yeast and mold count test. We also do endotoxin testing. And I'm going to be talking about another one of the tests that we offer, which is also a cell culture performance test. So thank you for asking, yes.
Thank you.
So the last part of the topics that I wanted to actually cover today was some actually variability that we saw in cells by unknown species that were in poloxamer 188 lots. So even after I told you all of this, and I share with you that there are, of course, some degrees of differences in different kinds of poloxamers just because of the wide use that they have in the marketplace, customers, of course, continue to use what we call regular grade poloxamer in cell culture applications. And the problem with that is that, well, there's not a lot of support that we can give around to customers around that because those poloxamers are not fit for purpose. They're not designed for cell culture. But we do remain committed to supporting customers using the Kolliphor P188 for this application. And during the summer of 2021, we actually have a few customers reach out to us because they were having some issues with some regular grade poloxamer in their systems. And what they were seeing was that there was a reduced BCD, viable counts of cells. And so we asked them to please send us samples of that batch to our lab. And we actually started doing the testing. So let me actually introduce you to our lab. So our lab was established early 2021, and both it's actually a space where we have facilities to do cell culture, small cell culture work, and formulation work as well. And the idea of the lab is not only to, of course, first and foremost, to support customers and by doing experiments and where we release application data to better understand how our ingredients behave in bioprocessing. And I mean, and it's actually just the commitment of that BASF is putting forward to understanding not only the shear protection, right? Because poloxamer 188 is only one of the surfactants that is inside your tank, right? There's other surfactants, for example, like your antifoams. So we are committed to actually understanding those surfactant systems, and our lab is actually the main tool that we're actually doing that with. So in our lab, and Phil mentioned some of the tests that we actually offer customers when we sell them Kolliphor P188 bio. And one of the data points that we offer our customers is results from a functional cell culture test that we do in this lab in Tarrytown. And here on the right is actually an example of how that dossier, that letter, looks like when we give it to customers. And essentially what we do in this cell culture test is test two conditions. One is a high shear stress worst case condition. We understand that poloxamer 188 is used as a shear protectant. So we are trying to replicate really high worst case shear forces in a flask, right? So all of these are done in flasks. And then the second condition that we test is actually a condition where we try to concentrate any other impurity that there may be in the poloxamer. So as I mentioned, we have the analytical test that we do to test for hydrophobic species, but customers raise the question, well, what is there something else? So we actually set up this condition to actually concentrate those other impurities that we don't know as much about. And we know that impurities, even at very small concentrations, can actually have a really hard effect on disturbing that shear protection that poloxamer 188 can offer, as I explained earlier in our talk. And what are the things that we measure? So we measure, of course, total cell number, percentage of viability, BCD, and cumulative and specific growth. We offer this. If this test is not validated, we are working on validating. We're setting up our team to actually get the test validated. It's not part of our COA. However, it's another data point to support our claim that this is our product that is fit for cell culture. And we offer it to customers upon request for every single lot of P188 bio. So what is the data that we saw? So customers came to us. They said that they were seeing some problem in their tanks, right? And as I mentioned, our test is a flask test. But we were able to replicate the customer's complaints, right? So here on the left, our assay is a 4-day assay. And here on the left, I have the cell viability numbers on the y-axis. X is the numbers of days. And if you look at yellow was the customer sample that we received that was underperforming. And our positive control here is a bio lot. Our negative control is a lot of, actually it was a lot of regular grade poloxamer that had been identified and flagged as something that is problematic in cell culture. And of course, we do also another negative control, doing a hydrophobic spike to actually simulate those conditions. And here you can see that the yellow line, which is the customer sample that our customers sent us, as they actually saw in the bigger tanks, it underperforms in our test as well. And when you look at the BCD, the same story is true. That when compared to our positive control, the cell density is also lesser. So our test was able to actually capture and replicate some of those results that the customer saw in bigger volumes. So if you actually are experiencing problems in your system, we are able to use actually our knowledge and our facility to actually maybe help you sort those out. And just in wrapping, I just want to reiterate that we at BASF are very interested and we have a compromise to actually build that knowledge of shear protection and understanding of the surfactant systems in bioprocessing. We are the leading manufacturer of poloxamer 188, and our bio offering, our Kolliphor P 188 bio offering, is the one that we actually support for cell culture, because it gives you all of the shear protection, minimizing the risk of having variability and having it impact negatively your process. I also wanted to share with you that we are expanding our biopharma ingredients team, not only with people, but with facilities like the Biopharma Ingredients lab. In this lab, we, as I mentioned, we perform application studies and technical studies, not only in cell culture, but also in formulations, to try to understand better how our ingredients are used and how can they be optimized in your process. So please put our knowledge in manufacturing and polymer chemistry to your use. And thank you very much for your time. I appreciate it, you attending today.
Thank you, Nadya. I just wanted to remind the audience that if you have any questions, we have a few, but wanted to remind everyone to please submit questions within the Q&A inbox, which is part of the webinar structure we have here today. I did have a few questions, Nadya. First one is around the lab. If you can share more information about what we can do with customers to support their applications in our laboratory, things that we can do.
Absolutely. So as I mentioned, one of the bigger uses that we're giving the lab this year, customers who had some negative experiences with regular grade poloxamer 188 last year we are actually supporting their conversion to our Kolliphor P 188 bio product. And we understand that those management of change can be, oftentimes it's very involved, and it involves a bigger team on our customer side. So we actually are providing all sorts of different application data for the customers. For example, data between any kind of variability in batches of bio from one batch to the other. That is something that we are generating for some of our customers. We are also interested in understanding, for example, we understand that many of our customers use silicone-based antifoams. And everybody uses a different one, and they have their different tricks. We also are supporting some customers and actually understanding how the poloxamer 188 and those antifoams may be working with or against each other in a situation, in a tank situation. So, those are some examples of the kind of work that we are doing to support customers. And as I mentioned, we have a vast knowledge in polymer chemistry. So, if you think that there is another question in your bioprocess that involves the poloxamer that maybe you want answers to, we are very open to actually collaborate with you and figure out your problem.
Thank you. And then, do we think that switching to Kolliphor P 188 bio from the standard pharma grade would negatively impact protein expression or bioprocessing?
So we have, so with the product launch in 2016, there was a lot of customer interactions, right? So a batch of customers received samples, and they had really great positive feedback on the way that the poloxamer performed in their bioprocess. We have not received in the years since then, so the launch was in 2016, we have not received any customer complaint regarding performance in their process. So we think that that there should not be any real impact in productivity and yields when changing from regular pharma grade to Kolliphor P 188 bio.
Is BASF measuring trace elements?
BASF is measuring trace elements. So we understand that there is a big concern on the trace elements that could be part of our different raw materials. We did an initial measurement of trace elements in some batches that we manufacture in 2019 and 2020. We're going to continue to actually do that analysis also in batches, more recent batches. What we found was that, so we measure calcium, magnesium, manganese, zinc, and iron. And what we found was that everything was in very small, actually most of them except the iron, because we make the poloxamer in a stainless steel reactor, are the only trace elements that are present in our poloxamer, and they are in the parts per billion amounts. It varies depending on the batch, but it's in very small amounts. And this is upon request for customers. So if you have questions regarding a particular lot, let us know, and we can actually provide that for you.
Good point. Thanks for bringing that up. What are some of the other common impurities observed? Is there any one any one that is a cause of concern or impacts cell viability specifically?
So we definitely know about the hydrophobic species causing a direct impact on the viability of cells. However, any kind of species that would be more surface active than P188 would actually cause a problem in that shear protection, right? So in the case of the hydrophobic species, they're more hydrophobic than P188, but anything that would be more hydrophilic than P188 would also be a problem. So we think that, I mean, if you are in the, so that's what I mentioned initially, that P188 is this kind of Goldilocks kind of example, and where it was just enough. It has enough ethylene oxide and enough propylene oxide to offer that shear protection. And anything that varies from that ratio, from P188, will actually cause some disturbance on the shear protection.
Okay, we have another question. What could be the frequency of addition in the monoclonal antibody process, and what concentration should be added if stock is 10% solution?
So it actually, that question varies. It is very dependent on two things. One is the cell type that you're using, and also the kind of process you're using. So for my customers that use fed batch, the concentration that seems to be the most used in those systems is between 1 to 2 grams per liter of poloxamer in those systems. However, I also have customers that are trying to actually do continuous manufacturing, right? So they use more intense production settings, right? So for perfusion, for example. And those, I have had conversations with customers and where they use anything between 4 to 8, even up to 8 grams of poloxamer in those more shear intensive processes. So that is something that it would be trial and error on your particular process.
Thank you. Is BASF's new lab able to measure protein expression with you?
So I will say that our cell culture capabilities are much more advanced than our protein and formulation capabilities. So the formulation and protein part of the bioprocess is something that we are building, and we are actually currently hiring for. So that for now, no, we cannot do that yet, but it's on the plans.
And regarding the cell culture test that we're in the process of validating, we anticipate that to be on the C of A in the near future. I know it's something that we've been working on for a little while. We expect that to be a release criteria on the Kolliphor P188 bio-CV in the near future, or continue to list it as upon request for the results that we do with the testing that we do in the lab.
Our goal, the goal is to actually have it as part of our COA, but we also recognize that for us, especially inside BASF, it's actually an undertaking to actually change some of that release criteria. So the first step that we're taking is actually validating the test. We've during this past year that we had the lab open, we have done a lot of optimization on the method that we're using because our scientist is both a cell culture and polymer chemistry expert. So she actually has been very diligent in actually improving upon a few of the parameters that weren't optimal. So we actually have been generating quite a bit of data with our latest method, and we are hoping to actually validate it in Q2 of this year. And then the process to actually have it as part of our COA will commence internally in BASF. But yes, our goal is to actually make it as part of our COA.
And the next one, sorry.
Oh, let me make a clarification about the cell culture test too.
Sure.
Even though it is not validated and it's not part of our COA, we do our functional cell test before releasing a lot of P188 bio. So at the same time that we send samples to do our HPLC, they send samples to my lab to actually do the testing. And if those two tests are not, they don't look as they should, we will not release that lot. That will trigger our internal investigation for us. So I wanted to actually make that clear to the attendees that is something that we do before we do actually do the release of the lot.
Thank you. And another question is, different customers have different subclones. How does BASF confirm that our product can satisfy different customer requirements?
So you are absolutely right. Whoever asked that question, and we recognize that the clones that we use in our lab, which are, as I mentioned, show S cells, are not necessarily representative of what you may have in your production line. But we do, what I can tell you is that as we have continued to grow the team in biopharma to try to understand more the shear protection, we have come to understand better how the chemistry of the poloxamer and how for example, the molecular weight distribution in a poloxamer can affect the cell performance, for example. That's something that we have been looking at closely. And what we have found is that our bioproduct in the molecular weight distribution that it offers, it seems we have tested several, so we have tested CHOs, we have tested HEC293s, and we're testing other cell types. And we have found that it continues to actually offer the best shear protection to those cell types as well. But we do recognize that, of course, that your subclones may have different sensitivity than the ones that we use for testing in our lab. But we're trying our best that is why we are investing so much time and resources in trying to understand that divot and where the chemistry meets the biology to try to actually offer a product that is fit for purpose, yeah.
And then how does BASF manage security of supply for Kolliphor P188 bio? How do we manage making sure it's there for customers?
So we produce our poloxamer in our Geismar, Louisiana site. This is a multi-product. Production site, and we are offering. We are actually within that plan. Kolliphor P188 bio has priority on manufacturing, and we actually, so that actually gives us the edge over some other products in our portfolio to actually continue to actually have a higher level of safety stock of the Kolliphor P188 bio. So that is, we understand that this is a critical ingredient, and we take that, several precautions to actually have secure safety of supply for our customers.
And I think you covered this in an earlier question, but I'll just ask you again in case I missed something. Is it possible to provide the cell types specifically sensitive with shear stress?
Cell types that may be specifically sensitive to shear stress?
Right.
That is actually something that I don't have comments on, mostly because we test. Of course, we do not develop our own cell lines in our lab, and we don't have access to any clone of any kind that actually makes any kind of pharmaceutical or any kind of biologic pharmaceutical. So I don't have any comments on that. But we can have a conversation if you're interested in if actually talking to us will be beneficial to maybe get to the bottom of that question.
And another question is, What is the main reason that poloxamer 188 affects the total cell number? I find the same poloxamer 188 has different performance for different customers.
So the specific, as I mentioned in the seminar, poloxamer 188 happens to have the perfect ratio of ethylene oxide and propylene oxide to actually offer that protection, right, by embedding itself, absorbing itself into the cell membrane. When you actually have, and with the Kolliphor P 188 Bio, we actually have manufacturing controls, analytical tests, and functional tests on cells to actually make sure that the performance is the same in your process batch to batch. With other grades of poloxamer 188, that is not the case. There is no testing of any kind, which is what can lead to the variability you see in your process. So if you get a regular pharma grade and you put it in your process, one batch can absolutely be the best performing batch you ever saw, and then the next batch you buy can actually have something in it that impacts the shear protection, and then you actually see that reduced viability and productivity.
Okay. And another question is there a specific recommendation for the use of Kolliphor P 188 Bio in microcarrier culture? The question continues that we're facing some problems with microcarriers adhesion to the vessels due to foam.
So that is an excellent question, and you're not the first one to ask me regarding microcarriers. So that is something that I would be very interested in, if you person asking the question would be interested in having a conversation with us so we can understand your system better. I would be very open to having that conversation. Microcarriers are not equipment that we have in our lab, so it's something that we don't have direct access to actually experiment on. But we will be very eager to have this conversation in trying to understand what the problem is and trying to ideate around solutions on better use of the poloxamer 188 in your system.
Okay. And another question, if we have literature about the poloxamer 188 affecting the shear protection properties of the poloxamer. So I think we have quite a lot of literature around that, don't we?
Quite literature of P188 being a shear protectant?
Yes.
As a shear yes, there is vast publications on that. And several of that we actually have also done our internal modeling in BASF to actually model that behavior. And of course, there is tons of primary literature where that has been tested well over, yes.
And another question is, are we working on a liquid formulation product?
No, but let's talk.
I was going to say, I didn't think we had one. I know we just had the solid
powder. No, we do not have one, but let's talk.
And can we comment on any of the degradation of poloxamer 188 as it reaches its expiry date or retest date? And if we have any information that would provide more information around that impact on the production of cell culture.
So our poloxamer 188 has a protectant, an antioxidant called BHT added to it to actually help it with the oxidation, the normal oxidation that it would actually, I don't want to use the word suffer, but have throughout its life. But we find that when the poloxamer is kept in cool conditions that are not terribly humid, that its life is actually its shear protection life is actually pretty long. So if your product is not performing as well, let's talk.
I think there's specific stability indicating factors, as you said, oxidation, pH, molecular weight, that are part of the C&A and the product spec that can be evaluated as well to ensure as your product's getting closer to that retest date, that those can be tested to ensure that you're not having the problem, that the product's not producing.
Yeah, that is true. Actually, we have actually data all the way to, is it 3 years, Phil, that we have retest data?
Yep. So we do have stability data for 36 months.
Yeah, and we can actually provide that upon request.
Yep. And then there's one last question. Is it possible to have too many cells in cell culture media that you want, so that you don't want to add poloxamer?
Is it? Then that is actually something that you would have to check with your formulation, right? So your media formulation, right? Because having a lot of cells means that you have to have a lot of other things in order to actually keep your cell populations high, right? You have to have the right amount of amino acids, the right amount of trace metals, the right amount of sugar to keep that up. What we have found is that, as a rule of thumb, cells that are grown in suspension systems, so if in the absence of serum, right? So if you are using chemically defined media, traditionally the rule of thumb is that you need a surfactant to help with the shear protection, and that is what the job of poloxamer 188 does. If you actually can have some kind of critical mass of cells within your system to not need poloxamer, that is actually something I don't know. I think at the end, you will certainly have to also evaluate not only all of the things that you will have to put into your media in order to try to achieve that, but also all the output, right? So what is the concentration that your system can withstand of the amount of MAB you're making, for example. So that is something I have not tested, I have to say.
And that seems to end all of our questions for today. Thank you, Nadya, for a very engaging presentation and a great Q&A. I think that wraps up the webinar today, and I'd like to thank everyone for attending, and thank you for your questions. If there's anything that we've missed, please feel free to reach out to Nadya or I from BASF. And we'll also review the questions that have come up in the Q&A and make sure we're able to get back to people on things we might not have addressed. And thank you very much for attending.
Thank you. Have a great day.